Okay, thanks Kavita and Deepa for inviting for this workshop. So what I am going to talk about today is my take on evolutionary dynamics. I mean, you may wonder, as a computer scientist or as a theoretical computer scientist rather, why I am suddenly interested in evolution. Okay, this is not something which is considered, um, you know, in fashion. But recently, there are more and more computer scientists actually who are looking at evolution. And there are a couple of reasons for that. So the first reason is, of course, as computer scientists who over many years have built up lots of tools and techniques to analyze things like Markov chains, dynamical systems, we feel very confident that we can make a lot of biology and evolution rigorous. So that's one point. However, in that attempt, we are beginning to realize that there are many problems in our own discipline which we can take inspiration from nature and evolution as such and make progress on some of our core issues. So even though as a computer scientist, somebody may or may not be interested in evolution as such, but the kind of problems that I have seen over the last few years that show up many of them actually seem currently beyond the techniques that computer scientists have. And so for me, this is a great test bed for understanding what we can and what we cannot prove. And today what I'm going to do is to talk about a very, a very basic evolutionary dynamical model, something which is probably classical, and consider the problem of trying to understand how quickly it reaches its steady state. So this will be a stochastic process or a Markov chain in this setting. And what we will try to understand is how quickly it reaches its steady state. And using some of these techniques, or rather developing some of the techniques to analyze Markov chains, we will be able to give some answers, or at least for a large class of evolutionary dynamics, we will be able to prove that the convergence is rather fast, which is what biologists knew probably 100 years ago, but we just joined the party. Okay, so this is what the talk is going to be about. I mean, and this is joint work with Yanis Panagios, who's a PhD student at Georgia Tech, and Piyush Srivastav, who's a postdoc at Caltech. Okay, so let me start with telling you what the motivation is, since there are probably a large fraction of people here who care about biology. And this is a problem that I worked on a few years ago, was to try to understand viral evolution. So this is a cartoon of what really happens. I guess for many of you here, this is not something which is so interesting, but let me quickly take you through it anyway. So we are trying to model the life cycle of a viral cell and how and what it does when it comes in contact with a T cell in the human body. So there are many stages. So you see here, this is the cell trying to enter this object. It uses its proteins to make an entry. It has some proteins, in particular reverse transcriptase, which converts its RNA into DNA. This process is not very high fidelity. There are some errors here when this kind of replication happens. This DNA then goes on and integrates with the DNA of the host cell, which is replicated, and this is a very high fidelity process. And then the viral material is again assembled by ribosomes and so on, and things come out. So this is what you can see. So this is a process which we are trying to understand a few years ago. And we went back to some of the most basic evolutionary dynamical models that were there. This is what I'm going to tell you. So in case you want to keep in mind, there will be no sex in this talk. I mean, sex is still difficult for computer scientists. So we are going to talk about asexual evolution of this type. Okay, and let me just point out that in this phase, in this diagram here, three important things which characterize evolution are selection, which happens, so different viruses, different virus strains have different abilities to penetrate a host cell. It depends upon the shape of their proteins and so on. And this is the mutation step, where when the virus is trying to copy itself, it makes errors. In fact, in some sense, that's why it is using the host cell, because its replication is not so great. And yeah, and this is where it comes out. So even though you may have a single genome, it can give rise to a large number of closely related genomes. And a lot of these viral, antiviral drug design strategies try to increase this mutation rate somehow of certain strings. And this is, so trying to understand what happens in steady state in this process is very important. And indeed, these were the simulations that I did with some chemists and biologists. Okay. So, so the kind of models that we will look at will be these classic Wright-Fisher type models, which came in population genetics in early 1900s. I mean, it's kind of a moot point, but I would still like to mention that these population genetics models have applications far beyond biology. you can model a whole host of phenomena, for example, social processes, language evolution, advertising, and so on, using this. Because after all, evolution is nothing but how information moves in a constrained space. So this is a very general phenomena, and models have wider applicability than just population genetics. This is always a good thing to keep in mind, especially if you are young and looking for applications. Now, what we would be interested in, as I said, is that this is a process. It's an iterative process. Evolution is an inherently iterative process. There is a sense that it reaches a steady state. How quickly does it reach the steady state? So we want to understand the time it takes to reach steady state. Okay? We will define all these things quite formally in a little bit. And why are these questions important? Okay, so from a person who does simulations, these questions, I mean, how do they handle it? What they do is they track parameters of the population, let's say different types of genomes and so on, and wait for these parameters to stabilize. This is, while a good idea in practice, it's not really theoretically justified. I mean, one can construct examples quite easily where you can see the statistics stabilize for some time, and then you're still far from being in a steady state. So especially when things like when you're using these models to design drugs and all, you need to be very sure what is really going on, and whether you are whether the population steady state matters or not. And this kind of steady state behavior ties up with the previous talk in terms of error threshold and so on. I'm not going to talk about it, but so driving the virus past its error threshold is why we want to, so we want to understand all these things. Okay. And I think a little bit more philosophically, at least for me, it matters what the steady state is, because we'll get to the math in a little bit, but we'll see that the state space, the number of possible states in which the system can be really large, actually. And what we want to understand is how quickly is this state space explored in a certain sense. And this, this is important from the point of view of how quickly life could have evolved. I mean, after all, if you go to the prebiotic world, processes like this are what were happening. And unless they stabilize relatively quickly, there is no way we could have built more and more higher structures on top of them. Yeah. So the size of the state space will be, we will come to that, and we would want to understand how quickly the steady state of the Markov chain is reached with respect to the size of the size of the state space. So the size of the state space will be typically very, very big. Okay? So imagine that you have a genome. Now each genome, if it's a binary sequence and the length is L, the size of the steady state is 2 to the L. And if L is a billion, then this is 2 to the billion. Right? So clearly, we didn't arrive here by exploring all these possibilities. Right? So understanding how quickly the process stabilizes is very important, given that we know or we have a sense that the state spaces can be typically very huge. Okay, so let me give you an example to be very concrete about the model. So imagine that we have two types, one in 0 and 1, and we have a population of size n. And this is a process where, it's an asexual process and it's an error-prone process, so we have mutations. So we capture this mutation by this matrix, let's call it Q, which is described by, in this particular example, by a single parameter, mu, which stands for mutation rate. And think of it as the probability of going from 0 to 1 or from 1 to 0. So when copying happens, what is the probability that 0 becomes 1 or 1 becomes 0? Okay, so this is the mutation probability. Different types have different fitnesses, which again we will capture by numbers, integers in this case. So, and what this gives rise to is a Markov chain whose state space is in this case, very simple. It's just pair of integers which add up to n. Okay, so S and T are non-negative numbers which add up to n. So the size of the state space here is n plus 1. When you have two types, the size of the state space is just the size of the population. Okay, so here's an example what could be happening inside how this thing is evolving. So imagine that at time t, the state of the Markov chain, which is now, so five types, 3 are red and 2 are black. Okay, so in the reproduction step, or each red multiplies with a fitness, it has a fitness, let's say, of 2, so each red ball gives rise to 2 red balls, and each black ball gives rise to 1 black ball. This step is not stochastic in these models. What is stochastic here is selection. So out of this intermediate pool, which is in this case 8 balls, you select 5 at random. Okay? Now, because we chose replication rates differently for different types, this already induces the selection bias. There are other ways which you could do that. They all turn up to be equivalent. And then you also need to incorporate the mutation step where each of these types can mutate according to this mutation matrix. And this is again a stochastic process. And the chain continues. Okay? So this was the state of the system at time t, and this is now the new state of the system at time t plus 1. This is a random variable. Okay. So is the stochastic process clear? I see a lot of very young people here. The population size we assume is constant here. So this is 5, and this is really not happening. This is a mathematical slowdown of the process. What is really happening is from here directly to here. Okay, we are just slowing it down to conceptually separate the steps. The population size is 5 and remains 5, and this continues. Right. So we do the Wright-Fisher one in this case, where you just take n. Right. But you could also do Moran sampling. Yeah. I think all these models present the same sets of difficulties, which is what we are trying to understand, whether we have techniques to handle them or not. Okay. Okay. So. So let's talk a little bit about convergence and the mixing time. So this is something which probably many of you have seen many times, is when there is no mutation, we know that eventually the population becomes either all red or all black. This is called this process of fixation. And this corresponds to, in this language of Markov chains, as these two states being absorbing states of the Markov chains. Once you hit them, you never leave, you know. And people study here what is called the time to fixation, which is the hitting time. Okay, when the chain has absorbing states, you look at the hitting time of starting from. So what is the hitting time? You can start from anywhere, and you look at the maximum time that you start from the starting point to the state that you want to hit. However, when you introduce mutations or maybe variation for other reasons like sex and so on, the chains start to become ergodic. And ergodic just means that now no state is absorbing. You will never get trapped anywhere. You can move from anywhere to anywhere. Okay, and this is how things are really. In this case, you can the fundamental theorem of Markov chains tells you that if the chain is ergodic, then there is actually a unique steady state, which is which can be very complex actually. Okay, so it's no longer the case that we can just analytically write down, given the parameters of the Markov chain, what the states, what this state is going to be. And what is the mixing time? So the mixing time is the following quantity. It is the time it takes for you, no matter where you start from, to reach close to this steady state. Of course, you will never reach this steady state. Okay, this is unless you start precisely from the probability distribution, which is the steady state, you will never reach it. So you can only talk about reaching close enough. Okay, and this closeness is measured by this parameter epsilon. And the mixing time is defined to be the distance between the distribution at time t of this Markov chain to the steady state. So when this distance becomes less than epsilon, for the first time, that is what the mixing time is. Okay. So it's, yeah, if the chain had absorbing states, then this reduces to the hitting time, but this is a more robust and more general notion of reaching steady state. And this is what we will work with. And what our goal is to try to understand an upper bound, the mixing time. We want to give an upper bound on this. Really, there is not much you can do other than prove a mathematical theorem once you have a Markov chain and you want to declare that the chain has mixed. I mean, you really cannot, and this is very misleading, that you keep doing a stochastic process and look at samples and at some point kind of declare that, okay, the chain has mixed or it has reached a steady state. There is no way you can look at the population and predict that. If somebody knows a way, I would love to know that, but at least mathematicians and computer scientists have not been able to find one yet. Okay? So the only way to establish this is to mathematically prove that this is an upper bound on the time it takes to mix, and run your stochastic process for that much time and then stop. The nice thing about these numbers is that even if you do more than what is predicted, you're only getting a better sample, not a worse sample. So this property is not going worse with time. Okay. Okay. So the example I gave you was of two types, and you can wonder, what is the big fuss about? There are n things. So how does it matter? N states, and that looks kind of relatively easy. Okay, so let's go to a more general setting, which is of multi-types. Now we have types from 1 to m. Okay, and instead of having a 2 cross 2 mutation matrix, now we have a m cross m mutation matrix. And again, fitnesses are now given for every type, sigma in 1 to m, and this is what is called the fitness landscape. And we capture both Q. Q is a stochastic mutation matrix, and we capture these fitnesses as a diagonal matrix, which I call A here. N is the population size. So what I presented to you earlier was two types. So this is just a generalization of that. The model remains the same. And in particular, we have random variables, which I use x sigma t. So this is the number of types in the population at time t of type sigma. Okay, so this now is a random variable. And we assume that the population is held to a constant, which is n. And the Markov chain, again, is a generalization of what we did in the first or second slide. So you have this population, each sigma produces a sigma copies of itself. So this is what the intermediate number looks like. Now you have this huge pool. You pick n at random from here, in the Wright-Fisher sense. And you can also, while you're picking, or in any of these steps, you can mutate each of them. But now you do a mutation according to this matrix Q, and this is the stochastic. Now let's look at what the state space here is. Okay, so if you have m types and a population of size n, and even for modest numbers like m equal to 30 and n equal to 10,000, you start getting numbers which are bigger than the number of atoms in this universe. Okay, all three numbers here are real, okay? So in fact, the experiments that I was doing with HIV was something like this. And this is the number of, this is the size of the state space, okay? So now it's kind of, the numbers start to grow in your face and kind of say, okay, how could the process be reaching a steady state so quickly given the state space is so high? So what was known about it? So motivated by this work on HIV with Narendra Dixit and Piyush Srivastav, in 2012, when we, yeah. Right. So think of m as 2 to the L. Okay? So I'm already picking a very low number for it. Yes. Okay? So already as you see that these numbers are very small. I mean, not all genomes are out of this 2 to the L. You can already make that simplification. They are not all different. So maybe 30 of them are still different fitnesses. Okay, so we proved that, in fact, the mixing time can be logarithmic in the state space size. So that is the key word. So you want to prove mixing times which are logarithmic in state space. And I mean, just a little bit, okay, so maybe I should say that we proved something, but it was under an extreme restriction of parameters, in particular, those weak selection and very strong mutation, which kind of never made sense, even for the application that we were looking at, okay? Yeah. Right, so this is missing here. So what it should be really is something like some function of m times log n. Okay, so what I'm going to write down is just highlight the dependence on n. Okay, and m are just absorbs as constant. There is also mu and q and all that stuff which is not written. Right, right. So, okay, so the question of proving mixing times in Markov chains which are logarithmic, yes. How do I calculate? So this is a combinatorial problem. So you imagine that you have n balls, and they can be one of m colors, okay? So what is the total number of configurations you can have? So that is something like n plus m choose m. And for m which is not as big as n, something smaller, let's say even less than n by 2, that asymptotically behaves like n to the m. So that's the right calculation for that. Okay, so let me make a small remark here about computer science and why these things are important. So for many problems in computer science, there are algorithms which are designed by implementing a Markov chain. This is the only way to do things. For example, if I give you a graph and I want you to, let's say, count the number of matchings in a graph, okay? So the graph is given to you. It's a small object, but the number of matchings in the graph, I guess a matching in a graph is a set of edges which don't have endpoints in common, okay? So just ways to match people together. You just imagine that. So that number can be really big. And what computer scientists typically do is design a Markov chain where each step of this Markov chain is efficient. You can go from one matching to another matching quickly. Same as in evolution. You can go from one state to another state quite cheaply. There is, that computation is inbuilt in how things are happening. What is not clear is how to kind of get around this exponential space so quickly. And in computer science, logarithmic versus non-logarithmic makes the difference between something like p versus np, okay? So proving such results that the mixing time is logarithmic in the state space is extremely, extremely important. And when I saw first these examples from evolutionary dynamics, I thought this is a good way to try to check if what we learned in computer science designing algorithms is actually useful. And we proved some results, but they were not at all close to what you would expect. So in the case of two genotypes, and let's say the two types have the same fitness. So this is what is called the neutral setting. Okay. Here, this is a simple exercise in probability to show that the mixing time is something like log n, where the base depends upon the mutation rate. So if the mutation rate is really close to 1, then this can be large. And if it is bounded away from, sorry, if it's very close to 0, it can be large. And if it's really bounded away from 0, then this can be logarithmic. So this everybody here probably knows. Another setting which probably people here know about, and it is often used in population genetics, is to assume, because we can't really prove things for all parameters, we just assume that the selection is weak and the mutation is also weak, and then move to what is called diffusion approximations. And there again, one can, once you move to this setting, which is mathematically rigorous, you can get some mixing time bounds again, but this is very restrictive, at least in the setting of viruses and so on. So last year, I proved the following result, which satisfactorily resolved the problem for the two-type case. Okay, so for the case of m equal to 2, and for all values of a and mu, I could get a mixing time which was which behaved like what you expect or what you expected to see in practice. So the question that remained was what happens for more than 2 types? What can we prove for more than 2 types? Okay, and that is what we really resolved in this paper that I'm talking about. That for all settings of evolutionary dynamics of the kind I described before, no matter what parameters you pick in the model, the mixing time is logarithmic in the state space. Okay. I'll come to this in a little bit. And so this kind of gives some evidence to me that, okay, so given that this could be a model for what was happening in the primordial world, this could be some evidence that evolution can happen fast, despite the fact that it has to guide itself through a very complex terrain of states and so on. And of course, it also makes all these computational strategies for designing drugs and so on based on simulating viral populations sound. But of course, everybody here never doubted for a moment that your simulations were wrong, right? So, okay, so for me, as a computer scientist, what was really interesting is that it took new techniques in computer science to prove these results. Okay. And that was something to connect, we connected dynamical systems, which are deterministic systems, kind of deterministic analogs of Markov chains, to Markov chains. So what and this is what I want to show you in whatever time I have, is how, even though you have a stochastic process, you can obtain rigorous results about it by looking at the underlying dynamical system. And this seems like a very promising connection, and it should be useful in not only in evolution, but in a fairly general setting, in particular in trying to understand algorithms in computer science, which are based on Markov chains. Okay, so in the next few minutes, I'm going to talk a little bit about the proof, or roughly the proof strategy. The proof is kind of fairly technical, but let me tell you at least what the guiding principle is. So just recall that in our setup, we have these random variables, x, sigma, t, which tell you how many types of sigma are there in the population at time t. Okay, so here is where the dynamical system comes in. Okay. So imagine you did the following. So x of t, you can also think of as a vector, because a vector with m coordinates, each coordinate for each type. And suppose you fix that vector at time t, and you computed the expected population at time t plus 1. So this is a mathematical notation which tells you the expectation of a random variable conditioned on what happened at time t. You're conditioning at time t and looking at the expectation at time t plus 1. Okay? So this is something you can actually calculate quite explicitly for this Markov chain. And it turns out, it's not a big surprise, it turns out to have a very nice form. It is really just applying the matrices QA to the current population and then normalizing them to have unit mass. Okay? So you can also think of this as some kind of an infinite population version of these dynamics. And this is a dynamical system that we will look at. Okay? So remember that because the population size was n, the vector xt by n lives inside the simplex of m dimensions. And the output of this is also a vector of length m, which lives inside the probability simplex. So the function f gives you a dynamical system from the simplex of m dimensions to m dimensions. So this you can also, as I said, think of as the infinite population limit of this dynamical of this Markov chain. Okay. Now, what can we infer from f? So one thing we can infer from f is that f captures the expected motion of the Markov chain. So something really complicated is happening in this huge space, which has state space n to the m. This is a very small dimensional space where all I'm keeping track of is of what is happening in a very very simple way, just the expectations. And what will turn out to be the case is that this function will allow us, or rather, when looking at the limit points of this function and the geometry around it, it will allow us to talk about the mixing time of this huge Markov chain. Okay. So in particular, for this Markov chain that I described, one can quite easily show that this dynamical system has exactly one stable fixed point inside the simplex. And why is that? Well, if I keep applying this operator f again and again to a point, it's the same as applying this matrix QA again and again and again. Okay? So when you apply a matrix again and again to a vector, it converges to the largest eigenvector. So this is a Perron-Frobenius theorem from non-negative matrices. So for this dynamical system, we know everything about it. I mean, we know no matter where you start from, you will always hit this steady state point, V. And yeah. So how does one prove mixing time bounds for Markov chains? So there is no one technique. Okay, so there are many ways to try to understand the mixing time of a Markov chain. One particular method that we will also use is called the coupling method. So coupling method is just a very general strategy. It says the following, that so imagine doing the following thought experiment, that you have two Markov chains of the same, two copies of the same Markov chain. So you have xt and yt, which tell you what the states are. These are two copies of the underlying Markov chain. And suppose you could do the following. Suppose you could construct a coupling between them. So what is a coupling between two Markov chains? It's essentially a correlation between these two objects, so that if I look, so each one of them independently notices no difference whatsoever. Okay. However, the two chains start to come together in some manner. Okay. So this you can imagine mathematically doing. I'm not going to tell you more about it here. It can already take a lot of time. But this is a general strategy where if you can construct couplings and show that the two chains collide in time t, then you can bound the mixing time. So this is some basic theorem in the theory of mixing time. Of course, the difficulty is in coming up with the coupling. I give you two populations and I want you to, okay, so what is it that I really want you to do? So this one will mutate, this one will also mutate. I want to couple mutations, I want to couple selections so that the distance between these two populations is reducing over time. And none of them is really noticing that anything fishy is going on. So this chain and this chain should not notice that anything different is going on and what is usually going on. But you, as someone who's sitting on a higher plane, can kind of control these two things together. So for example, I'll give you, so what is coupling? So suppose I promise to two of you that I'm going to give you a sequence of coin tosses, right? So what I could do is the following. I could toss a coin, and whatever I see, whether it's heads or tails, so let's say I tell it to him, and I tell him the complete opposite of it. Right, so I am keeping my promise that he gets a sequence of fair coin tosses which are independent in each iteration. He also gets the same, but they don't realize it that they are getting they are correlated, completely correlated, and this is the magic that happens in these couplings. That this process is running as it was supposed to run. That process is also running as it's supposed to run. However, in this particular case, by the way I have constructed things, they will never ever see the same coin, because if he sees heads, he sees tails, and if he sees tails, then he sees. Okay, so the mathematical key is to kind of use this function f, the dynamical system that comes out as the expected motion of the Markov chain, to construct this coupling. Okay, and here we use stability properties of the fixed point along with certain measure concentration phenomena to show that in a short amount of time the two chains can collide. Okay, so the next few slides are I just told you I will do a coin toss here, and I see whatever I see. I tell this person, if I see a heads, I say heads. If I see a tail, I see, and to him, I will just say the opposite. So both of them get, whatever they are using these coin tosses for, they are getting fully independent true sets of random bits. But between them, they are not independent. So it could be a very general mechanism. So there is, it doesn't have to be. I just give you an example of a coupling which is not an independent coupling. A trivial choice for me to do is to do two coin tosses and just keep one reserved for him and one for him. But I'm trying to be clever and trying to kind of make sure that these two guys never end up meeting, right? And so I'm kind of, their behavior is random from an outsider point of view if one person looks just at them. But once you look at them together, the behavior is no longer random. And this is allowed. And this is the magic of, this is the magic of coupling. And we have, this kind of boils down to trying to understand that if the chain is mixing fast, what are the forces? Why is it mixing fast? What are the driving forces? And coming up with a coupling is kind of the same as identifying the governing forces. What is driving them close to the steady state? And how do you use this? So mathematically, you would use this by saying that, okay, we will start one Markov chain as wherever you want to start. The other one, I will start at the steady state already. So because they are coming close to each other, the steady state never moves. So because this one is coming, so this one will eventually come close to the steady state as well. So the designing the coupling is designing the trying to understand what is causing these chains. So this is a very general methodology to prove mixing time bounds. However, in a specific case, you have to come up with a coupling. That's the hard part, how to couple them. So what is stability? What kind of stability am I talking about? So this is, for example, a simplex in 3 dimensions. And this is maybe one fixed point here. So by being a unique steady fixed point, trajectories from everywhere are converging to this. And the stability of a fixed point in a dynamical system is governed by what is called the Jacobian of the dynamical system. And in particular, if the maximum eigenvalue of the Jacobian is bounded away from 1, so if the absolute value of the maximum eigenvalue is bounded away from 1, then the system shows stability. Okay, so in control theory, this is what people mean when they say a notion of talk about stability. And in fact, if you were to try to understand the rate of convergence of dynamical systems, it is really this number, rho, which determines the rate. How quickly are you going towards the fixed point? Okay, so there are a couple of observations. So one quick observation is because we are sampling n balls, we have enough concentration. There is enough independence still in the process, and you can show that this function f will bring two. So here you don't have to do anything. Okay, no cleverness required. In a short amount of time, even constant time, you will come very, very close to this fixed point. Okay. So you will come about 1 by root n distance to this fixed point. Now is really where the game starts. Okay, so why do the populations come closer? If you want to prove a bound on the mixing time using the coupling method, it will require you to drive down the distance between xt and yt, not to 0, but to something which is less than 1 by n. This suffices. Okay. Once the two populations come around 1 over root n close, because of all this stochastic noise, they can just keep kind of doing strange things around. They can diverge and then come back again, and all kinds of pathological mathematical behavior that can happen, one has to rule out. So this is where stochasticity kind of hurts you, and this is where the fact that there is an underlying dynamical system helps you. Okay, so what we can show is we can construct a coupling using this function f, which has the following property, that in one step, the distance between the two chains in expectation shrinks by this factor. So this is the one-to-one norm of the Jacobian. It's a technical quantity, don't worry about it so much. Just think of this as some kind of a Lipschitz constant for this function. Okay. So if we could show that this number here is smaller than 1, then we would be done. However, this, okay, so you can do that when there are two types, okay, and this can give you another proof of the result I proved in last year. However, you can show that this number can be bigger than 1. Okay, and the intuitive reason is the following, that suppose you have a vector and you rotate it a little bit, then the L2 norm of it doesn't increase, but the L1 norm of this vector can increase. This is something I would let you figure it out. So this can happen. And the next thing that we use is to then, instead of, so in one step you cannot hope to come close. Okay. What we then do is to take a large number of steps and then hope that we can couple these things together. And this requires a lot of technical details, which I'm going to omit. But let me just point out that even though in one step the Jacobian's one-to-one norm could be bigger than 1, you could show that because the fixed point is stable, there is some constant k for which the one-to-one norm of the kth power of the Jacobian is less than 1. And this will allow us to give the coupling. There is a lot of things you need to do, and the technical core is to handle all this stochastic noise. It's a lot of work mathematically. Yeah. Yeah, so because we are looking at a rather strange norm, which is one-to-one norm. So what is this norm? Okay, so when you have a matrix A, so when you typically talk about the norm of a matrix, you say the maximum over X of the 2 norm. You're looking at how the matrix changes the length of vectors when you measure the starting length in 2 norm and the ending length in 2 norm. This is what typically people study. Okay, when we talk about one-to-one norm, we want to look at how it behaves, how it changes the 1 norm of vectors. Because we are working in probability spaces, we need to look at this ratio. And this can be very different compared to the 2-to-2 norm. However, the fact that the largest eigenvalue is less than 1, which is the same as saying that the 2-to-2 norm is less than 1, implies by Gelfand's theorem that there is some value of k for which the one-to-one norm of the kth power is also less than 1. Okay. Okay, so using all this, what we can show is that for these k steps, even though there may not be contraction in one step, in k steps there is contraction, and provided that the stochastic noise kind of remains controlled. And this is anyway I'm not going to talk about. And the situation becomes harder and harder to show. So I mean, as you come closer to the singularity, yeah. So these forces that we are guiding them with together is the coupling. No, this all experiment is going on in my head, right? So we are trying to understand what are the forces which are driving this system too. Right, yeah. Okay, so the effect of noise kind of becomes larger and larger, and we need to use a multi-stage argument. How the distance changes, so up to log n by n, there's a different argument. Then, once you come close to 1 by n, there's a different argument. And then finally, once you reach close enough, you can show that chains will collide in one step. So there are several stages to this argument to finally prove the result. Let me just conclude by saying that, okay, so as I think you pointed out, that I'm hiding the dependency on M here. It's not like I'm trying to hide something, it's just an ugly dependency at this point. I think trying to improve the dependency on M would be really a very important problem here, okay? And trying to get these, so there are still mathematical barriers, there are still theoretical barriers to doing that. And well, trying to do this, trying to understand this problem is, I mean, for me, a very interesting way to develop techniques in Markov chains. Finally, I would also like to say that we don't know how to handle the case when f has multiple fixed points. And this, for instance, happens in at least a couple of situations, which is when you introduce sex. And there are also models in social sciences like evolution of language and so on, where there can be multiple stable fixed points. Now what happens? Now this theory kind of breaks down, and we need an extension to this theory. So this is a challenge mathematically. And finally, yeah, I guess this is probably more relevant to a computer science or mathematics community, that this connection that we have found between dynamical systems and Markov chains can actually be much more useful for understanding Markov chains, even though you're probably not, you may or may not be interested in the mixing time, maybe some other thing, but next time you see a Markov chain, try to compute this dynamical system and see if it can give you any intuition about the Markov chain itself. And with this, I would like to stop and thank you.